What's up, TUMNAD lectures? In this video, we are looking at what we call chemical digestion and absorption of what we call carbohydrates. If it's your very first time to watch me, please make sure you like the video, comment down in the comment section, and subscribe. Let's go ahead and get started. In this video, we are going to be mainly focusing on what we call chemical digestion and absorption of what we call carbohydrates in great detail. But before we go into the detail of today's discussion, I want to first take much little of the time and we see the anatomy of this diagrammatic illustration because it is super very important. Understand how do we call all these portions before we start off the video. Okay. Now, when you see the whole of this kind of diagrammatic illustration, it is going to be showing us right from what we call the oral cavity down to what we call to the small intestine. It's all that simple. Now, what happens is that this one here is going to be like the oral cavity, okay? The whole of this dilated structure here is going to be what we call the oral cavity. And then also going to be having this small invagination here. This one is what we call the stomach. I have drawn it in a very small way because we are not going to be having all that significant amount of the chemical digestion of carbohydrates in what we call the stomach. That's why I have drawn it very small. And then this one here, this big portion here, this one here is what we call a portion of the small intestine, which we call the duodenum, the jejunum, and then the ileum. And then the last one, this one here, we have just drawn a very single kind of the absorptive cell, which we call the enterocyte. Okay, so now this one is going to be our enterocyte, this is going to be the luminocyte, and this is going to be the basolaterocyte, which is going to be facing the blood vessels. The luminocyte is going to be having a lot of different kind of the brush border enzymes, which we shall see later as we are proceeding in what we call the videos. Okay. And then, of course, we're also going to be having some of the contents which are going to be coming from the different tubes from what we call the accessory digestive structures. For example, what we call the pancreas, the gallbladder, all those structures, they are all going to be emptying their their secretions into this the small intestine through what we call the sphincter of Oddi. Besides that, we also have what we call the accessory digestive structures that pours their contents to the oral cavity from these these structures here, which we call the salivary glands. Okay. So now here we have come with this this big piece of what we call the sweet potato. It's very nice, it's very delicious. But we want to first digest this this big piece of potato, and we see which kind of the carbohydrate are we going to be getting from what we call this the sweet potato. Which kind of the carbohydrate are we going to be getting from what we call the sweet potato? The most one that is super much significant, we are going to be getting what we call the starch. Okay, we are going to be getting what we call the starch, because it's the commonest carbohydrate which is going to be gotten from what we call the plants. Now, the question is, what are those the polysaccharide molecules that make up what we call the starch? There are two main types of the polysaccharides molecules that make what we call the starch. If you have noted watched the previous videos, we looked at all these the molecules that make up what we call starch, and we looked at what we call the structural makeup plus all the bonds that you're going to be forming, that you're going to be having in those those structures. In this video, we are going to be slightly a bit faster because we already have an idea in the previous video. If you have not watched that video, please go and check that video so that you become so much used with this these bonds. However, we have two kinds of the molecules that make up what we call the starch. The first one is this this long chain of the polysaccharide, which is going to be having a lot of different kinds of the monosaccharides that are going to be linked together. This this long chain of the polysaccharide, which is not going to be branched. This one here is what we call the amylose. Okay, this one here is what we call the amylose. Super very important. And then the second of the molecule that make up what we call the starch is this this long chain of the polysaccharide, which is going to be having a lot of different branches. You know how this this molecule is called? This one here is what we call the amylopectin. Okay, this one here is what we call the amylopectin. Okay, these are the two molecules that are going to be forming what we call the starch. Okay, so now in this video, we're going to be looking at these of the molecules, how we are going to be breaking them down. But besides that, when we talk of chemical digestion, we are mainly focusing on enzymes breaking specific types of the bonds into the absorptive forms, for this case, which we call the monomers. Now, the question is, what are those types of the bonds that are going to be linking these of the monosaccharides in amylose and then amylopectin? In the previous video, in amylose, we said they're going to be having this kind of the bond, which is going to be formed by what we call carbon number 4 and then carbon number 1. We called that bond as a alpha 1, alpha 1 to 4, glyco, glyco, glycosidic bond. Okay, that is the name of that bond. And this kind of the amylose, it only has one type of the bond which we are calling alpha 1 to 4 glycosidic bond. It's all that simple. Okay. Now, when we come to the amylopectin, this one is somehow a bit different because it's going to be a long chain of polysaccharide having a lot of the branches. It has two types of the bonds. The first bond is this kind of the bond which is going to be on what we call carbon number 4 and carbon number 1. And this type of the bond is what we call alpha 1 to 4 glycosidic bond. Okay, and then also we said that we also have another kind of the bond which is responsible for the branching of this kind of the amylopectin. This kind of the bond is going to be on carbon number 6 and then carbon number 1 on another kind of the monosaccharide. This bond links this kind of the monosaccharides together, making a branch. And this kind of the bond is what we call the alpha 1 to 6 glycosidic bond. Okay, this one here is what we call alpha 1 to 6. Alpha 1 to 6 glycosidic bond. Okay, so now those are the two main kind of the bonds that are going to be linking the different monosaccharides that form what we call the amylopectin. Okay, now having seen now the bonds that are making sure that these kind of the monosaccharides are held together, let's go ahead and see how we are going to be chopping them down. Okay, now the question is, what is that kind of the enzyme that's going to be breaking this kind of these ends or this kind of these bonds? Now we're going to be having this kind of this structure here, which is going to be part of the parotid, the submandible, the sublingual. Those kind of the salivary glands are going to be providing us what we call saliva, which is rich in an enzyme. You know how that enzyme is called? That enzyme, which is going to be coming from this structure, which you're calling the salivary gland, into the oral cavity through various ducts. This kind of enzyme is what we call salivary alpha amylase. Salivary alpha amylase. That is the name of the enzyme. Now the question is, this enzyme, which kind of the bonds is it going to be targeting in this kind of these molecules, the amylose and then amylopectin? Because we have two types of these bonds. Now, this kind of the salivary alpha amylase is only going to be targeting what we call the alpha 1 to 4 glycosidic bond. It's all that simple, okay? So now this enzyme, which is like a molecular scissor, you know, enzymes are like molecular scissor, they aim at cutting different types of the bonds. But this salivary alpha amylase is only going to be breaking alpha 1 to 4 glycosidic bond. Now it will come over here and then it will come over here on this bond and it breaks it. It comes over here, it breaks it. It comes over on this amylopectin, it breaks it. Okay? So now in that way, it is breaking only 1 4 glycosidic bond. Now, does it have effect on alpha 1 to 6 glycosidic bond? There is not any kind of the effect of alpha salivary amylase. It means that this kind of the bond, it has its own specific type of enzyme which is going to be breaking it in the process of digestion, especially in what we call the small intestine. It's all that simple, okay? So now, having broken different types of these bonds, what are some of those kind of the molecules are we going to be forming after the chemical digestion of carbohydrates in what we call the oral cavity? At the back of your mind, you have to know that this kind of the chemical digestion, by the effect of alpha salivary amylase, is going to be very, very, very small percentage. Approximately 10 to 20% of chemical digestion is going to be happening in the oral cavity by alpha salivary amylase. Okay, meaning that you're going to be having some other enzymes that are going to be doing a biggest percentage of chemical digestion of carbohydrates. Now, once this chemical digestion has been done with what we call salivary alpha amylase, we are going to be forming a lot of different substances which are categorized into two. We're going to be forming what we call the disaccharides. We are also going to be forming what we call the oligosaccharides. Okay, and when we talk of disaccharide, it means those are going to be two monosaccharides which are going to be held together. And when we talk about oligosaccharides, they are going to be 3 up to around 9 or even 10 monosaccharides held together. Now, what are those kind of disaccharides that we are going to be forming following the chemical digestion of carbohydrates within the oral cavity? We are going to be forming the following. Now, The first one, which we are going to be forming, is going to be this disaccharide, which is going to be held together by this kind of the bond we are calling alpha 1 4 glycosidic bond. And this kind of disaccharide is what we call maltose. Okay, is what we call maltose. Is what we call maltose. And of course, maltose is going to be having what we call a glucose molecule held together by another glucose molecule. Okay, so now that's the first kind of the disaccharide which we are going to be forming. The second one is going to be what we call this disaccharide, which is going to be having this kind of the sugar, this kind of the monosaccharide which are held together, and this one is going to be what we call a glucose molecule, is what we call the galactose molecule. So now this kind of disaccharide having a glucose molecule and the galactose, this one is what we call the lactose. Okay, this one here is what we call the lactose. Super very important. Okay, and then another one that is super very important is going to be this molecule, which is going to be containing this kind of the glucose molecule with another kind of the monosaccharide, which are held together by 1,4-glycosidic bond. And this molecule is going to be having what we call glucose and what we call fructose. Now, this kind of disaccharide is what we call the sucrose. Okay, this kind of disaccharide is what we call the sucrose. Super very important. Now, we also have now what we call oligosaccharides. Oligosaccharides are going to be having three kind of the monosaccharides linked together and above. We have the following. We have this molecule, which is going to be having three kind of sugar molecules that are going to be linked together. Okay, this kind of the oligosaccharide is going to be having glucose molecules only, which are going to be linked to one another. So now, how do we call that kind of the oligosaccharide having only sugar molecules? This kind of the oligosaccharide is what we call maltotriose. Okay, is what we call maltotriose. It's all that simple. Okay. And then the last one that is super very important. Remember, we also have some kind of the molecules here that are going to be having still what we call the alpha 1 glycosidic bond that are not yet broken. Okay, so we have that kind of the big pieces of that kind of the molecule. Now, that kind of the molecule, I want to draw it in this kind of the simplest way. For example, I'm going to be having a lot of different kind of the monomers. Okay, they are going to be linked together. But again, you're going to be having other kind of the monosaccharides that are going also to be linked together, okay, which are going to be linked together in this way, okay. So now you realize that this kind of the molecule or this kind of the oligosaccharide is going to be having this kind of the monosaccharide linked together by different types of the bonds. This one here is going to be what we call the alpha 1 to 4 glycosidic bond, but this one here is very much special. This one here is going to be what we call alpha 1 to 6 glycosidic bond. Now, this kind of the molecule, this kind of the oligosaccharide, which is going to be having this kind of two bonds, you know how this one is called? This one here is what we call alpha limit dextrins. Okay, what we call alpha limit dextrins. Okay, so now these are the molecules that you are going to be forming following the breakdown of salivary alpha amylase. However, they are going to be a small amount. Okay, they are going to be having the biggest percentage of the different types of these molecules which are not going to be broken. It's going to be very small pieces of these molecules. Okay. Now, when these molecules now move from what we call the oral cavity, they move down to what we call the pharynx, they move down to what we call the esophagus, and now they move to what we call the stomach. Now, within the stomach, we are not going to be having a significant amount of what we call chemical digestion, because once the enzyme salivary alpha amylase reaches into the stomach, it becomes inactive by the influence of high levels of what we call hydrochloric acid in the stomach. So we're not going to be having a significant amount of what we call carbohydrate digestion chemically in what we call the stomach. Okay, now these substances are now going to be pushed in what we call the small intestine. Now, in the small intestine, we have these cells here. These cells are super very important, and these cells, they are sensitive to what we call high levels of oligosaccharides. You know how these cells are called? These cells here are what we call the eye cells. These cells are super, super important. Whenever they become activated by oligosaccharide, they release a molecule called cholecystokinin. Okay? They release a chemical called cholecystokinin. Cholecystokinin is a very good, important kind of the hormone that's going to be flowing through the blood. It moves and then it goes on this kind of this specific structure, which is part of the accessory digestive structure. You know how this structure is called? This structure here is what we call the pancreas. And within the pancreas, we have a specific type of the bulbous structure, which we call the pancreatic acinar cells. What happens is that cholecystokinin has receptors on what we call pancreatic acinar cells. It will stimulate pancreatic acinar cells to release one of the most important type of the enzyme. You know how this enzyme is called? It is going to be having a lot of the vesicles having a lot of different type of enzymes. But the enzymes of interest that are responsible for breakdown of what we call carbohydrates is what we call pancreatic amylase. So now this kind of the pancreas is going to be giving us what we call pancreatic amylase. Now, what happens is that this pancreatic amylase will be moving through what we call the main pancreatic duct and then comes over here, this kind of this sphincter. You know how this sphincter is called? This sphincter is what we call the sphincter of Oddi. What happens is that cholecystokinin has receptors on the sphincter of Oddi. It come over and bind on these receptors here. You come over and bind on these receptors on the sphincter of Oddi and make it relax. Once it relaxes, our molecular scissors we are calling the pancreatic amylase will come and then drop inside what we call the duodenum. From there, they'll all start now cutting more and more of what we call the amylose and the amylopectin into more and more of these different kind of the disaccharides and also what we call the oligosaccharides. Now, something which is unique is that the pancreatic amylase, it also only targets alpha 1 to 4 glycosidic bond. It's all that simple. It means that the effect of what we call pancreatic amylase is going to be the same as salivary alpha amylase. However, the effect of what we call pancreatic amylase is going to be more and more significant compared to what we call alpha salivary amylase. It's all that simple, okay? Now, having now made a lot of this kind of the oligosaccharides, what we call the disaccharides now, let's go ahead and see how are we going to be breaking them down into their final absorptive molecules, which we call the monosaccharides. That's what we want, okay? Now, when we come over on this kind of this area, we're going to be having this kind of this cell which we call the enterocyte. Okay, this kind of the cell is what we call the enterocyte. And enterocyte is what we call the absorptive cell. Now, let me first cut over this kind of the enterocyte. We zoom it over here and see exactly what we are trying to mean. Now, this enterocyte is going to be having finger-like projections. Okay, it's going to be having finger-like projections, which we call the microvilli. Now, these microvilli, these finger-like projections are going to be having a lot of this kind of enzymes. You see this kind of this blue, red, green dots. Those are what we call brush border enzymes. It has a couple of different types of brush border enzymes. These are the enzymes you are seeing on the luminous side of this enterocyte. Okay. Now, let's go ahead and see each of these kind of the brush border enzymes and their effect on different types of the oligosaccharides and even also on what we call the disaccharides. Now, this kind of this enzyme, part of what we call brush border enzyme. This one here is what we call maltase. Okay, this one here is what we call maltase. Now, this maltase, guess which type of the disaccharide is going to be breaking? This one is going to be breaking what we call maltose. What happens is that maltose will now be running to this kind of this enzyme which we are calling the maltose. Now, what happens is that maltose is going to be running to this enzyme. So now we are going to be having this maltose which is linked together by specific type of the bond. Now, what happens is that by the effect of this maltase enzyme, it will break over this kind of the disaccharide into individual monosaccharides. And what are these monosaccharides? These monosaccharides will have what we call glucose and even also what we call glucose. It's all that simple, okay? Now, I also have another kind of the brush border enzyme. This brush border enzyme, this one here is what we call lactase. Okay, is what we call lactase. So now, guess which kind of disaccharide is going to be running to lactase? Lactose. Okay, so now we come over to this lactose. It will move and it goes on this structure on this enzyme we are calling the lactase. So now, when this kind of the lactose comes over, on what we call lactase enzyme. This lactase enzyme will break this kind of 1,4-glycosidic bond, and then leading to the formation of these two types of the monosaccharides, which is going to be the glucose and also what we call the galactose. It's all that simple, okay? Then we also have another kind of the brush border enzyme that is always complex to one another. Super very important. This kind of the brush border enzyme is going to be one of the most super very important. It has one of these components which we call sucrase. Okay, which we call sucrase. And then it also has another one here down, which is super very important. You know how this enzyme here is called? What we call isomaltase. Okay, is what we call the isomaltase. So now let's go ahead and see what does these enzymes, the isomaltase and sucrase, what do they do exactly? Okay. So now, what do these enzymes break exactly? Now, this kind of the enzyme which you're calling the sucrase, what is going to be breaking? We still have what we call sucrose. So now, what happens is that sucrose will come over and move to this kind of the enzyme which we are calling the sucrase. So now, this kind of the sucrose is going to be containing what we call glucose and what we call fructose. What happens that a sucrase will come over and break this kind of the disaccharide into individual kind of the monosaccharides, which is going to be what we call the glucose and even also what we call the fructose. Okay, so now we have also separated what we call sucrase. Now what happens is that we also have what we call isomaltase enzyme. What are those kind of the molecules that this enzyme is going to be targeting? This enzyme is going to be only and only targeting alpha 1 to 6 glycosidic bond. It's all that simple. This enzyme is going to be targeting alpha 1 to 6 glycosidic bond. So now this molecule is also going to be coming over here. It's also going to be coming over here like this, going to be having a lot of the branches, like this. So now what happens is that this molecule is going to be having alpha 1 to 4 glycosidic bond, but it also has what we call alpha 1 to 6 glycosidic bond. What happens is that this isomaltase will come over and break that kind of the bond, and also breaks that kind of the bond. Okay. So now by doing that, it is only leaving alpha 1 to 4 glycosidic bond. What happens is that this kind of the molecule can come over on this enzyme which we are calling this for further what we call breakdown of alpha 1 to 4 glycosidic bond. It's all that simple. So now the final products, we are going to be forming a lot of different monosaccharides. These monosaccharides can be what we call the glucose. It can be what we call the glucose and also what we call another kind of the glucose molecules. Okay. So now the question is, this kind of the malt, how is this malt going to be also chemically broken down? We also have what we call maltase. Maltase can come over and break this kind of the malt into individual monosaccharides, which is glucose. However, this kind of the sucrase can also have a small effect on the breakdown of what we call malt. It's all that simple. So now, what are the monosaccharides have we formed? We have formed the following monosaccharides. The first one which we have formed is what we call the glucose, and then the second one which we have formed is what we call the galactose, and then the last one is what we call the fructose. Okay. So now, what have we formed? We have formed what we call the glucose. We have formed what we call the glucose. Okay, we have formed what we call the galactose, and then also we have formed what we call fructose. Okay, these are the three main absorptive molecules of what we call carbohydrates. Now, the question is, they are within the luminal areas. How do we absorb them into the enterocyte? It's all that simple. Now, within the luminal side of what we call the enterocyte, we have a lot of different channels that are going to be allowing the uptake of what we call different kind of the monosaccharides. What is very unique is that this kind of the monosaccharide which we are calling the fructose is very mean. He doesn't move with anything. He moves with on his rod alone. What happens is that we have this kind of the specific channel on the luminal side of this kind of the enterocyte that allows the movement of this kind of the fructose down the concentration gradient. Now what happens that this kind of the molecule we are calling the fructose is going to be moving, and then it passes through this kind of this channel, and it enters into what we call the enterocyte. So now we have now brought the what we call the fructose into what we call the enterocyte. So now the question is, how do we call this kind of this channel? This channel here is what we call the GLAT5. Okay, that's the name of the channel that allows the fructose to enter into the enterocyte. It's all that simple. So now the question is, how do we take in the glucose and then the galactose? It's very simple. Whenever cell on entire human body, we always have this kind of the primary active kind of the channels, which we call the sodium potassium ATPases pump, that are always maintaining what we call concentration gradient. Now, on this kind of the enterocyte, we have this kind of the sodium potassium ATPases pump. What this kind of the channel is doing, it is always pumping out 3 sodium ions and then bringing in 2 potassium ions. What happens that the levels of the sodium ions within the intracellular environment of the enterocyte is going to be decreasing. Okay, so now there is going to be a decrease in what we call the sodium ions within the intracellular environment of the enterocyte. Now, what does this one mean? It means that we are going to be having a lot of the sodium ions within the lumen compared to the intracellular environment of what we call the enterocyte. Now, by doing that, sodium would want to move down the concentration gradient into what we call the enterocyte. How does that one happen? We have this channel here. This channel here, what happens is that it allows the movement of what we call sodium to enter inside what we call the enterocyte, because this pump is actively pumping out sodium, causing a deficiency in sodium ions. So now what happens, because there's too much of the sodium ions in the lumen, it now allows the movement of this kind of the sodium down the concentration gradient. But what is beautiful is that sodium is very, very, very friendly and merciful. It helps some other kind of the monosaccharides to enter into what we call the enterocyte. Such a beautiful thing. So now what happens that this kind of the sodium, as it is entering, it comes over and then pulls on what we call the glucose. It enters with the glucose. Okay, it also enters with the glucose. It comes over on this kind of the molecule calling it the galactose. It says, hey, galactose, come, I can also help you to enter into this kind of the sweet environment, such that we also appreciate the beauty of how the inside environment of the enterocyte is very nice. Okay, so now by doing that, sodium has now helped glucose and galactose to enter into the enterocyte. It is such a beautiful thing. So now the question is, what is the name of this channel? This channel here is what we call sodium glucose co-transporter. Okay, sodium glucose co-transporter. It's allowing the movement of glucose and galactose to enter into the enterocyte. So now inside of the enterocyte, we have fructose, we have glucose, and we have what we call galactose. What is very beautiful that on the basolateral surface of what we call the enterocyte, we have a specific type of the channel that allows all of these kind of the monosaccharides to move out into the extracellular environment of this cell. What am I trying to mean? This kind of the galactose will now come over and pass on this kind of this channel. The glucose also come and pass on this channel, and then also the fructose this time around is also going to be allowing to move with others. It is now moving into the extracellular environment where we are going to be having a lot of different kind of blood vessels. So now these kind of the monosaccharides are now going to be entering this kind of these blood vessels. These blood vessels can be what we call the superior mesenteric vein, the inferior mesenteric vein. They'll drive these kind of the molecules into what we call another kind of the big kind of the vein, which we call the hepatic portal vein. From there, these kind of the molecules will now finally enter into what we call the liver. So we are going to be having now our fructose is coming there, our glucose is also there, and then finally the galactose. They have finally entered what we call the liver. Now the question is, what is the name of this channel that's allowing the free passage of what we call galactose, glucose, and fructose? This one here is what we call the GLAT2, okay, which you call the glucose transporter 2, okay? That is the name of that channel that allows the passage of what we call all the monosaccharides. It's all that simple. So now, Once the galactose and then fructose reaches to the liver, guess what happens? The galactose and then the fructose will get converted into the glucose. Okay, it's such another beautiful thing. And of course, the liver is going to be using those kind of the monosaccharides for the process of what we call glycogenesis, and even also what we call even glycolysis. Okay, so all going to be happening in what we call the liver. Okay. So now, TUMNAD, we have talked a lot of things. And if you think whatever I have talked is making sense, is adding to you, you are benefiting, please, you know what to do. Hit the like button, comment down in the comment section, and subscribe. Please subscribe. Thank you very much for watching. TUMNAD, as always, until we meet next time.